Okay, so let's talk about bacteria in particular. So bacteria are single-celled organisms, right? They don't typically have organelles like Golgi apparatus and endoplasmic reticulum and all these organized areas like an animal cell does. They don't have a nucleus, but they have what's called a nucleoid, which is an area where chromosomes and DNA gather. Okay? They do have a cell membrane, uh, and around the cell membrane is a cell wall. Okay, and that will be important coming up. So bacteria have a cell wall, and we're going to talk about it a bit more when we're looking at ways that we can stain bacteria and recognize what kind of bacteria they are. Now, when you see the bottom statement there, a cell wall around the membrane made of flagella, well, flagella is basically just a tail that can move back and forth to allow the bacteria to move. And peptidoglycans are just basically proteins, peptids, and glycans are sugars. So mixtures of molecules that have proteins and sugars that make up this cell wall. Okay, so here's another image of a bacteria where it has a nucleoid, it's got some ribosomes, it has a cytoplasm. So there's some similarities to an animal cell, but not that many. So it has a plasma membrane, which we covered in detail when we were talking about human cells or animal cells. And then right around the entire bacteria is a cell wall. Okay. This one in particular has a capsule around it. And bacteria that are encapsulated tend to be even more resistant bacteria, right? They have more ability not only to evade the host immune system, but they tend to be more virulent in nature as well. So this one happens to have a capsule listed, but if you just remember that in general, bacteria have a cell wall that surround the entire bacteria. Okay, so the movement is from the flagella or the flagellum, depending if it's singular or plural. What's chemotaxis? Well, chemotaxis is the term that we use for movement of organisms towards a substance. So there's some kind of a substance in, say, the tissue fluid, so a chemical. So a chemical is a substance. Taxis is moving. So someone last semester used the term taxi to remember chemotaxis. So you could use that to remember it. But it's basically movement of cells from one area to another. It doesn't have to be bacteria, any cells, towards a chemical that's attracting them. Okay, so let's talk about some details about bacteria. And we're really going to be comparing them to viruses. So bacteria are typically much larger than viruses. Okay? Viruses are tiny particles that, remember, need your cells to replicate. Bacteria can replicate or reproduce on their own. They don't need to use your machinery. The symptoms of a bacterial infection just depend on where the infection has occurred. So if it's in the lung, we would call it a pneumonia. If it's in the bladder, we would call it a cystitis. If it's on, say, the skin, we might call it a cellulitis. If it's in the brain, we'd call it an encephalitis. If it's surrounding the layers of the brain, called the meninges, we'd call it meningitis. So it just depends on where the infection is. Bacteria are often responsive to certain antibiotics. Luckily, otherwise we wouldn't have many effective treatments for them. Okay. How do we find out what kind of bacteria it is? Well, we can do gram staining, where we put a specific dye onto a colony of bacteria from, say, a sample we took from the blood or the urine, and different bacteria will stain a different color. Okay, so that's gram staining, and we'll talk more about that. Or we can take a sample of urine or blood or tissue and put it on a culture media and wait for it to grow. Okay, and if there's growth, we'll see what kind of bacteria that grew on the growth medium that we put it on, and what antibiotics is it sensitive to. So we'll actually get a report called a CNS, a culture of what the bacteria is, and an antibiotic sensitivity of that particular bacteria. We don't get these with viruses. We just get these with bacteria. Okay? So an example would be we took a swab of the back of someone's throat. We put it on a culture media to grow for the next day or two, and 2 days later we grow streptococcus. And we can also get a sensitivity report, meaning which antibiotics is this bacteria sensitive to. We can do the same for urine, right? Send off the urine for a culture and sensitivity. A couple of days later, we get it back. It tells us which bacteria it is and which antibiotics it's sensitive to. Okay? Some bacteria are sexually transmitted. Chlamydia, syphilis, gonorrhea. Mind you, some viruses are sexually transmitted. HIV is sexually transmitted. Hepatitis B is sexually transmitted. Okay, so it's not. That's just that. This one's not unique to bacteria. The overuse of antibiotics has created bacterial resistance in the population. Okay, so and this is our doing. This is our problem. Overprescribing of antibiotics kills off many bacteria, but the bacteria that have survived are resistant to those antibiotics. They can live despite being with those antibiotics. So they've developed antibiotic resistance. And that is a huge problem. And that's from overprescribing of antibiotics to patients that just have viral infections. They never needed an antibiotic to begin with. Okay? So we're being more and more careful with the prescriptions of antibiotics, not only because of the adverse effects that antibiotics can do to the individual, but we also know that as a population, we are causing very resistant bacteria to take hold while we kill off bacteria that aren't resistant at all. Okay? And we do not have a lot of antibiotics that are being created as the years go on. Okay? So that's an important factor to remember. Antibiotic resistance. Okay, so this is just a size comparison starting with a shaft of a human hair and then going downwards. And if we go to the bottom, you can see how microscopic a virus is, even compared to a bacteria, right? And then if you compare a bacteria to a red blood cell or a white blood cell, there's a big difference in size, okay? Okay, so when we're classifying bacteria, there's different ways of classifying them. Some of them are based on the shape of that bacteria. So cocci are typically just bacteria that collect in little balls, so we call them cocci. Bacilli are more rod-shaped, they're more oval in shaped. Spiral, as the name implies, they have a spiral appearance to them. So that's one way of classifying bacteria. The staining that we're going to talk about in an upcoming slide. So when we put a stain on the bacteria, it's either going to stain gram-positive or gram-negative. And we'll talk about that in detail in an upcoming slide. The size of the bacteria can help us determine what kind of bacteria it is. So I would say the most common groupings after the size and the staining and the shape, you usually end up with a description such as gram-positive cocci or gram-positive bacilli, or gram-negative cocci or gram-negative bacilli. Okay, so and then once we have one of those descriptors, such as gram-positive cocci, well, then we know that, for example, Staphylococcus or Streptococcus are in that group. So it really narrows down the possibilities once we can put them into one of these groups. Okay? And what's more important is once we put them into one of these groups, we know which antibiotics are most responsive and most effective to that group. Okay, so it really helps us to narrow it down. And you can get a gram stain in an hour or two. Okay, so culture and sensitivity can take a few hours to get. A gram stain, 1 to 2 hours, and we have it. And we have a description of what bacteria we're dealing with and what are the best antibiotics. Okay, and here's just some visual images of different shapes of bacteria. So spiral, cocci, such as Staphylococci, Streptococci, and bacilli, which typically look more like rods. They're more oval in shape. Okay, so let's go back to bacterial staining. And this is where we were talking about a very fast process of determining what group of does the bacterial infection fall into. It doesn't tell us for sure which bacteria it is. It just tells us very quickly, is it a gram-positive or a gram-negative? Okay, so gram-positive bacteria typically have a thick cell wall. So they hold on to staining very well because of how thick the cell wall is, and it absorbs the stain that's applied to them. So they're typically purple in nature. So when you look under the microscope, gram-positive bacteria, once we've applied the stain, are purple because they have a lot of peptidoglycans that hold on to the stain. Gram-negative have a relatively thin outer cell wall. So because they don't have as many of those peptidoglycans, they don't hold on to the stain very well, so they stain more of a pinkish. Some of the stain washes out very quickly. Okay, so very quickly we have gram-positive, gram-negative bacteria. Okay, does it tell us which one exactly it is in the group? No, but just by getting this bit of information, we will know which antibiotics they will be most sensitive to. Okay, so here's an image of both gram-positive and gram-negative staining. Okay, we can't we can't say for sure what kind of bacteria it is in the positive there, but we know it's gram positive. It stains positive. That breaks the possibilities into a much smaller group. Okay, same goes for the gram negative. So gram positive bacteria have a thick cell wall and a thick peptidoglycan layer that holds on to the stain much better. It takes up the dye, which is called crystal violet, and it keeps it that purple color. Gram-negative bacteria have a much thinner cell wall. The stain washes out easily, and they typically stain this pinkish color. Okay, so let's talk about bacterial culture and sensitivity some more. So a bacterial culture will help us find the specific bacterial infection that's causing the illness. We need body fluids or a swab or something that we can apply to the culture medium. So for example, if we think someone has a urine infection, we'll collect a urine sample, and then that will be put onto a culture medium to see if a bacteria will grow. And that would be the same for a blood culture or a sputum culture that someone has coughed up, or a wound culture. We can take a sample with a swab and apply it to a culture medium and wait for growth of bacteria. And this can be very important because prior to starting antibiotics, we would take these samples and determine the exact bacterial infection that's growing, and then that allows us to be very specific with our antibiotic choice. Okay, so really any fluid or sample from the body can be put on a culture media. And typically bacterial samples take 24 to 48 hours to grow. So it's not an immediate result like a gram stain, but it's more specific. It'll tell us the exact bacteria. The same would apply for a fungal culture, but fungal infections and fungal cultures can take weeks to grow. So for example, if we're looking for tuberculosis and someone coughs up a sample that in which we suspect tuberculosis, it can take many weeks to grow tuberculosis on a culture media. Okay? The sensitivity part of it is once a bacteria or a fungus has grown, antibiotics will then be added to the culture media to see if it kills the particular bacteria and which antibiotics the bacteria are most sensitive to. Okay, so a CNS is extremely helpful to help us direct antibiotic use, especially in very sick patients where we really need to determine which bacteria is growing. Now, I mentioned this earlier on in this module that some bacteria have a capsule around them, and I think we had an image of one. And encapsulated bacteria have a greater pathogenicity, meaning they have the potential to cause significantly more illness. And that's because the capsule can protect them from white blood cells and also many antibacterial drugs. Okay, so it also protects the bacteria from the immune system by avoiding phagocytosis, meaning cells of the immune system that try to engulf the bacteria and destroy it. The capsule can prevent that from occurring. Okay, so having a capsule is considered a virulence factor because it makes the bacteria even more difficult to clear from the body. I'm going to give you an example. A patient that has had their spleen removed, or they have lost their spleen from an injury and then it had to be surgically removed, they are at high risk for bacterial infections, especially from encapsulated bacteria, because the spleen is actually part of the immune system. It has a lot of white blood cells in it. So when a patient loses their spleen, one of the first things we think about when they come in with a fever is a bacterial infection causing a serious illness. And in fact, if you have lost your spleen, so we call those patients asplenic, they are immunized for as many infections as possible with vaccines, because we know that they are immunosuppressed just by the very fact of not having a spleen. Okay, so let's briefly talk about antibiotics and antibiotic resistance. Okay, so antibiotics are used to destroy or even just to slow down the growth of bacteria, depending on which antibiotic we're talking about. And you'll learn more of that in pharmacology. So that's the primary role of an antibiotic, is to either stop the growth of bacteria or to destroy and kill the bacteria. Now, overuse of antibiotics has led to antibiotic resistant bacteria. And we'll talk about that in a minute. But what has happened is that there's many bacterial strains now that exist that are extremely resistant to an assortment of bacteria. An example of one that you're going to hear of is MRSA, methicillin-resistant staphylococcus. And this bacteria can cause superficial infections on the skin, and it can cause deeper infections such as a MRSA pneumonia or MRSA sepsis, where it's in your bloodstream. And we know that this bacteria really doesn't respond well, or isn't cleared very well by antibiotics. Okay, so let's look at antibiotic resistance and how it occurs. So in number 1 on this slide, you'll see that there's an assortment of bacteria. Okay, and most of them are going to be sensitive to antibiotics. So when an antibiotic is applied to the system, say in the body, it destroys most of the bacteria, and the resistant ones remain. And they can thrive in the setting of the antibiotic being around. The resistant bacteria can then grow, and now you are left with resistant bacteria that can't be treated with that particular antibiotic. And there's some bacteria, such as MRSA, that can grow despite many antibiotics being in the system. Okay? And finally, those resistant bacteria can transfer their bacterial resistance to other bacteria. Okay? Meaning that now there's more and more bacteria that are resistant to an assortment of antibiotics.